Hi, guys. I'm yelling in my own ear. So, I don't see that anybody has joined the Hangout, which is absolutely fine. I see Holly in one chat window, and I have another chat window that says I have one viewer, but I have no idea who that one viewer is. So, if you wouldn't mind letting me know who's here one way or the other, so I don't think I'm sitting here talking to myself. That would be great. Um, what we're going to do tonight is we're going to focus on how to use NCBI in Genes and Consequences. And my hope is that this will primarily be a way to get questions answered. So if you've got questions or you've tried using NCBI, this is the time to get them answered. So I'm going to try to do a much better job of watching the chat windows. Because since I don't have people joining the meeting, I'll need some way to hear your questions. So I'd like to start by asking you guys, have you tried using NCBI with your kids yet? And if so, how did it go? Holly says her year is going well, but she is ready for a break. I bet she's not the only one. All right, so no questions have popped up yet. So I think what I'll do is just kind of walk you through the way that I use NCBI with groups of students using some of those same sequence that we use for genes and consequences. Again, I'm going to try to do a better job this time of monitoring the chat window. So if you've got questions, go ahead and put them up there. And if I don't see it right away, I will see it pretty quick. At least that's what I hope. So let me set up the screen sharing and let's go play with NCBI. All right, so this is the current NCBI landing page. It may or may not look like this when you go to use it with your students. This is one of the things that changes on NCBI quite incredibly frequently. So if you get there and it doesn't look like that, don't be alarmed, because the piece that you're going to need for genes and consequences is always going to be located somewhere on the front page, and that's BLAST. BLAST is what we're going to use. Now, if for some reason they move to popular resources and you don't see that on the page, you can always click this resource list A to Z, and BLAST is going to be under the B's. So I'm going to click BLAST. And this is another page that may change the next time you come in here. As a matter of fact, this one has changed a good bit from the last time I used it with a group of students. But what we're going to do for genes and consequences is we're going to do a nucleotide blast. So I'll click that one right here. And here's kind of what the screen looks like. So what we do with genes and consequences is we enter that sequence number. Now, students are going to actually do two blast searches for genes and consequences. They're going to blast the original sequence that was given to them on the electropharogram, and they're also going to blast the patient sequence. So let's see if I did this right. Oh, I did. So I just copied that actually out of the teacher's guide. This putting the sequence in here in this box where it's asking for a fastest sequence is super forgiving. If students put it in capitals with spaces every 3 letters, it's going to accept that and do the search. If they put the search in in all lowercase letters and don't put spaces in between it, it's going to accept that as well. It is really incredibly forgiving. One thing they can check, though, to make sure they've got their sequence in right is to make sure that they've got between 50 and 53 letters. The sequences in genes and consequences are all between 50 and 53 letters. One thing that's worked well for me with groups of students is to have them in pairs and one read out the sequence while the other types it in, and then the other one, the one that typed it in, reads it back to that one to confirm that they've got the sequence in there correctly. The next thing we're going to do is we're going to have to tell it which database we're going to look at. Now, what we want to do is we want to find this sequence where it's in a gene that's a human gene. And there are a couple of different ways you can do that. One is you can click Human Genomic and Transcript right here, and it's only going to search human sequences, and it's only going to search transcripts that are known. That's one way to do it. And the other way to do it is, let me unclick that. Unclick that. And go here. This is what the genes and consequences directions tell you. We need to look for reference RNA sequences. So that's only going to give us matches that hit in known regions that code for RNA. Then you'll get this sometimes. Oh, wait, that's an exclusion one. Because I took that human off, it's going to ask which organism we need to look in. And I want to look for humans. So if I put in human, I've got an option right here, but I'll actually get much better search results if I go with Homo sapiens. If I go with the right name. And then I scroll down, I'm looking for highly similar sequences. I'm not looking for dissimilar sequences. And I'm not looking for somewhat similar sequences. And then I'm going to hit the blast button. And then I'm going to get this. And have to do that again. But luckily, I had copied and pasted that sequence, so it didn't take too long. All right, there it goes. It's searching. And while it's searching, this search can take anywhere from a few seconds to a few minutes. Oh, that one wasn't too bad. But it's a good point to say, hey, any questions? Holly, it now looks like it may just be you and me. All right, so if questions come up or I'm not answering what you need or there's something on the screen that you're not seeing, let me know. All right, let me go back over here. Okay, so here are our search results, and it's actually it's aligned to 6 different sequences. This right here is going to tell me how closely my sequence that I put in matches the sequence that's in the database. And then here are all of the possible hits. Now, a couple of things. This query coverage tells us how much of the sequence we put in matches with the sequence that the database has returned as a close enough match. Now, you can see all of these are 100%. So that's good. That means that these variants, these transcripts here are all 100% similar to the 51, 52, or 53 nucleotides that I put in. Another number you've got here is something called an E-value. This is probably going to be way over the heads of your 9th grade kids, but what that is it's a number that is mathematically determined that tells you what are the chances that this match, that this sequence that I put in matches this variant happened by chance. Was it just random chance that those happened to match? And the E-value should be incredibly small numbers. So this is 3 times 10 to the negative 19. So that's a vanishingly small chance that this hit came by chance. So it's probably a pretty good hit. All right, quick check for questions again. Nope. Holly, you still there? Okay. All right. So once your students have found this, now they're going to want to go start finding pieces of information about this gene for answering the questions that are in Genes and Consequences. And there are two things that they can click. They can click the extension number right here, or they can click this description over here. And it's going to give you two slightly different results. So let's click this one first. So what I clicked was this right here, and what it does is it opens up a lot of additional windows down here. Now this window right here is telling us that it has matched the sequence that I put in to this gene right here, this Homo sapiens leptin receptor, and it's matched it to variant 6. It also matched it to variant I found up here, 1, 2, 3, 4, and 5. So there wasn't enough difference between the portion of the gene that we put in and any of these variants. It's still 100% match. Okay, so this is showing me that match between all of those sequences that it came back as a 100% hit. And you can see that one-to-one correlation between the sequence that I put in and the sequence of the gene that's there. Okay. So that does give us a chunk of information. Now there are other pieces that we can click on, like we can click this GenBank right here, and that's going to take us, maybe, there it goes. It's going to take us to an awful lot of information about that. Sorry, Answer and Holly. Going to take you to an awful lot of information about that gene. Now, if you're working with a group of ninth graders, this level of information is going to largely go over their heads. However, I want them to look at this and scroll through because there's some interesting pieces that they can begin to find in here. First of all, when we get to this point, it starts giving you links out to papers that have been published about the gene. And this is a paper that was published, as a matter of fact, just a little bit earlier this month. On the 2nd of December, just this month, we had a whole article about this leptin receptor and how it behaves in the plasma membrane. And that's kind of interesting that there are papers coming out about the genes that kids are looking on, and the papers are really timely and often, you know, even just the title of the paper gives you a little hint about what that gene does. But if we scroll down a little further, we're going to get to this summary entry right here. The summary entry is going to give them some critical information about what this particular gene does. Now, when kids start reading that, this protein is encoded by the gene in the GP130 family of cytokine receptors that are known to stimulate gene transcription via activation of the cytosolic stat proteins. Again, ninth graders don't need to know that, but when I get to this next sentence, this is a protein receptor for leptin, an adipocyte-specific hormone that regulates body weight. So that tells me something right there, that this is going to have something to do with body weight and fat metabolism. And then if we get down a little further, we've got this sentence right here that tells us that mutations in this gene have been associated with obesity and pituitary dysfunction. So it's a good idea for them to read this summary. No, they're not going to understand everything that comes out of that summary, but there generally is enough information in there that they can at least get a hint of what that particular gene does. Then we scroll down a little further and we can get some additional information. This right here is a list of the single letter amino acid abbreviations that make up this entire protein. Then I come down and I find some important things like the gene name, The abbreviation for that leptin receptor is LEPR. I can scroll down a little further. That's the sequence that I put in and checked, and it tells me exactly which exon that sequence came out of. Okay, so now I'm going to scroll back to the top, and I'm going to go back to my search results. And this time, instead of clicking this description over here, I'm going to come over here and I'm going to click this extension number. And it's going to take me to a slightly different listing for much of the same information. Actually, no, it didn't. It took me to the exact same place. So let me try clicking it again. There we go. So same information that we got to, just via another. Oh, we do have some additional information here. So this is showing me information for the whole gene instead of just that sequence that I put in. So I can see all of the exons. If I scroll all the way down, this gives me a complete list of all of the nucleotides, all of the A's and T's and C's and G's that make up that entire gene. They can look and see exactly how long it is, because I think that's one of the questions in Genes and Consequences. So there's nucleotide number 1, and we run all the way down to 5,101, and then there are a few A's left there at the bottom. All right, that's part of what I wanted, but not everything. Now, sometimes NCBI gets a little hinky, and it doesn't give me exactly what I'm looking for. It takes me someplace that's not exactly what I wanted. So there are a couple of things over here in related information that often can get you to the information that you're looking for. The 3 that I use pretty frequently are right here. Gene, and I'll show you what that gives you. Map Viewer, we'll look at that in a second. And then OMIM, which is Online Mendelian Inheritance in Man. Those 3 will generally give me all of the information that I need to answer the questions in Genes and Consequences. So let's click Gene and see if it gives me what I'm expecting. Ah, there we go. Okay, so this summary is exactly the same as the summary that we were looking at on the other page. The only difference is the font. But again, it's got those little key pieces of information in there that can give students clues about what this gene does. One of the other things that Genes and Consequences asks students to find is, where is it? What is its chromosomal address? And this section right here, genomic context, is what gives you that. This location right here, 1p31.3, is what they're going to need to record. Now what that means is it's on chromosome 1, it's on the P arm of chromosome 1, it's in the 31st band, subband 3. So that's what they're recording in their gene book. But if you scroll a little further, it gives you this nice little graphic representation of what's going on chromosome 1 and where that gene is. Now, the interesting piece about this is you can see other leptin receptor genes and other bits of sequence that are related to that. There's a huge amount of information here, some of which I understand and some of which I don't. Here's another list of articles in PubMed. Again, the titles of these articles will also give kids clues about what this gene does. These articles here are going to be about functionality of that gene, and some of those might be interesting little rabbit holes for students to jump into. But the next thing that you're looking for is this section right here about phenotype, because phenotype is going to tell you if there are any genetic disorders or diseases that are associated with the variant that we've put in, that the gene that we're looking for. They'll be listed right here. And here's the one that we know that that particular sequence is associated with, is this leptin receptor deficiency. Now, if you click that, You might hope that it's going to give you information about leptin receptor deficiency, and it will give you a little bit, but one of the things that it gives is a list of the tests, the clinical tests that are available to test for variants in that particular gene. And then there's a lot more information that goes along with that. So again, we've got some descriptions of the symptomology, some pieces of the puzzle for what this gene does. And I'm going to scroll back over here. Lots and lots and lots and lots and lots of information. Any one of these links, students could click that link and get some additional information. Again, if you're working with ninth graders, that's more information than they really need. And certainly more information than they're going to need to answer the questions that they've got for genes and consequences. But again, some of these are really interesting little rabbit holes to scroll down and see what's going on with those bits and pieces. Okay, so now I'm going to go back to related sites, and this time I'm going to hit the map viewer. And that is not giving me a map view for this particular gene. So I'm going to go back one more. And now click map viewer and hope that's a chromosome map. And while I'm waiting for that to behave, any questions, anything pop up, any ideas? Okay, so here's my chromosome map viewer. Here is a nice ideogram of chromosome 1, and this little red thing right here is telling me exactly where this particular gene is located on chromosome 1. And then if I come over here, the gene that I was looking at, the gene that I had a 1-to-1 match, oh, Holly had a really good comment. She says some of her serious students have trouble with this lab because there's not one definite answer to each question. Do they get frustrated when they do the scanning? Because that's one of the things that I've found with kids. Some of them attach to that, and I think students are much more likely to start clicking buttons until they find something that they recognize and can make sense of. And some of them tend to get really frustrated. Some of them will look at you and go, just tell me what to do. Just tell me what I'm supposed to write here. That's not uncommon. However, you know, we say it in GTAC, and it's absolutely true. There is value in the struggle. And especially those serious students, the ones that are really determined to get the right answer, they're going to pick up an awful lot of things from that scanning, some of which is going to be even more interesting than what they would pick up if you just told them the answer. All right, jumping back to map view. The sequence that we put in is highlighted right here so that I can see exactly where that is. But the interesting thing about it is I've got all of these other sequence pieces that you can look at. So these are all of the other genes that are located nearby in that particular region on chromosome 1. This is the same extension number that we saw before. So if I click this, it should take me back to this page, which should be familiar to me. And I'm going to go back. And then if I click the gene name in Map Viewer, it's going to take me back to this one. Okay, so we've got the basic answers to the pieces for genes and consequences. We found out what the gene does. We found out where the gene is. Again, if they blast the patient sequence, if they blast the patient sequence, it's going to take them to this same exact place. And now that I've got this phenotype, I have to scroll back down, this leptin receptor deficiency, Now there's another way to get some additional information. We can check two places. We can check OMIM, and we can search here in OMIM for leptin receptor deficiency. Did I spell deficiency right? Ah, there we go. Leptin receptor deficiency. Yeah, Holly says her students want the answer to just jump right out at them, and they get frustrated when it doesn't. That's not unusual. I think a lot of us are struggling with that same frustration that kids have. And if you think about it, we kind of train students to do that the answer is usually the word that's in bold in a textbook. And that's not what they're going to find when they start doing research of their own, or when they get to college, or when they're, doing searches like this for something for a job even. The answer may not be the thing that's in bold or the thing that's in the top of the page. But I think all of us are going to have to figure out ways to be patient with that frustration while we teach them something else and some other way to think about that. And OMIM is being real slow. Ridiculously slow. And just like kids get frustrated, I get frustrated when it takes too long to load. So I'm going to give up on that one and go back. Let me try clicking it one more time. If it doesn't load quickly, I'm going to go show you something else. Nope, still nothing. Okay, so what I'm going to do is I'm going to click this NCBI logo up here in the top, and I'm going to actually come back to this page. And now what I'm going to do is go to the genes and diseases section. So I'm going to have to click the resource list A to Z, and then I'm going to scroll down to the G's, and I'm looking for this one right here, genes and disease. There we go. And now I'm going to search for, leptin receptor deficiency. And I got nothing. But what is leptin receptor deficiency related to? Obesity. And I can search that one and find this information. That, look, leptin jumps right out at you. And there's a good bit of information in genes and diseases that's going to be related to that particular gene and what you're looking at. All right. So I went through that pretty quick. And Holly's got a point about kids get frustrated with that. So if you've got some ideas for how to tackle that with kids, share them with me and let me pass them along to Holly. If any other questions came up while I was flying through that, or you see something strange the next time you go to do this with your students, by all means, shoot me an email, because if I can't answer it, I'll just grab somebody else at the Institute that can. All right. So I guess we'll wrap this one up this evening. If any other questions pop up, send them to me. We'll do our best to get answers. And I think the schedule for the next one, I can't remember when the dates are, sometime in January or February maybe is when we have the next one of these. I'll send you a reminder a little earlier next time. But the plan is for us to use that meeting to talk about aluminum tolerant corn. And that means that the topic for the fourth virtual meeting is wide open. So if there's something in particular that you'd like to talk about, in particular, if we could get a whole group of us in here connected at the same time talking, Holly, that doesn't mean I don't think you're wonderful and I'm not tickled that you're here. But I feel a little bit like I've been sitting in my den talking to myself for a half an hour. If there's something in particular you want to talk about, let me know what that is. So aluminum tolerant corn is what we're going to dig into next time. We'll talk about how it worked, if you've had a chance to use it with your kids. If there are some hesitations or reasons you haven't used it, I'd like to hear that too. And if you've got any suggestions for what we can do to make that a better kit, we'd appreciate it. All right. Thanks for your time. Send questions my way. Have a good evening. Bye.